everyone. The transcriptomic research that I will be presenting today connects space biology with global health applications. I will be describing the radiation-induced roles of HDAC4 and MORC2A in relation to muscle cell senescence. I complete this project alongside Megan Sawant, Sophia Tran, and Dorina Nguyen. I will be presenting on behalf of my team, so firstly to introduce myself. My name is Jessica Wu, and I'm a rising senior at Troy High School in Michigan. I'm the president and co-founder of Guidance, where I coach about 95 students in computational biology and research methods to present at the University of Michigan. I am also the president of Science Olympiad, Biology Competitions Club, BioBuilder, and Red Cross Club. As a NASA's research intern last year, I worked with 3 other students to complete the transcriptomic and bioinformatic research that I will present today. I hope to continue my interest in biomedical research into medical school, and I'm striving to become a surgeon in the future. So why is space biology so important to understand, and how does it relate to my current project? Space biological research has a lot of connections to the well-being of people both in space and on Earth, promoting global health. Satellite communications allow people to engage with telemedicine, to improve equity among underserved communities through mapping medication distributions. Space biology has also resulted in many advancements in technology used in medical facilities today, including MRI and CT machines. Studying astronauts and other model organisms in space is crucial to further understanding the human body and providing insights into medical conditions here on Earth. The purpose of my research is to highlight suggested breakthrough within the space biology community regarding potential targets for cell senescent muscles. For a bit of background, ionizing radiation in space contains high energy which damages the cell and its biomolecules. The radiation can lead to the formation of reactive oxygen species, causing oxidative stress. Elevated levels of reactive oxygen species can cause DNA damage, muscle dysfunction, cell senescence, and pro-inflammatory signaling. Antioxidants, like nanoseria, which was introduced into our dataset, are molecules that protect against the reactive oxygen species composed of oxygen and cerium particles. In our experimental design, we will look into cell senescence, a state in which cells no longer divide, but they still release inflammatory chemicals that damage nearby cells. We analyzed dataset OSD426, aimed to test the protective role of nanoseria on oxidative stress in skeletal muscle cells. There were 6 sample groups present in the study that were RNA next-generation sequenced, each with a different combination of radiation, presence of nanoseria, and gravity conditions. We decided to investigate the effects of nanoseria on radiation's damage to cells, and thus analyze 2 different pairings. First, the gray pairing. Here we have nanoseria absence and radiation present in the flight sample. And second, the blue pairing, and here we have nanoseria presence, and radiation is again present only in the flight sample. Moving on to our preliminary data, this diagram includes the steps taken on the Galaxy platform for genetic analysis. We began by looking at PCA plots to explore the correlation of data. As you can see, the red and green samples are separated in both without nanoseria and with nanoseria groups, showing that there's a large variation between the exposure to radiation compared to on Earth. We then performed a go-seek analysis. Interestingly, we found a statistically significant biological process, response to external stimulus. We looked into this further through the Mouse Genome Institute database. Skeletal muscle tissue regeneration is a specific response in that process, showing that radiation has a harmful effect on muscle tissue regeneration that we need to look into. We analyzed the top 40 genes for the nanoseria present samples. We filtered them with the log4 chains of greater than 1.5, as well as a p-value of less than 0.05 as our significance threshold. In this volcano plot, we examined the differentially expressed genes in nanoseria present groups, with radiation being the independent variable compared. Our gene of interest is MORC2A. MORC2A regulates the part of the DNA damage response pathway, which occurs in radiation. So with the presence of the nanoseria treatment, there was less reactive oxygen species and less DNA damage, and therefore a downregulation of MORC2A. We then analyzed another volcano plot comparing radiation in space versus on-ground samples, except in this time with the absence of nanoseria. HDAC4 works in a similar way to MORC2A, as it's also part of the regulation process in the DNA repair pathway. So with the absence of nanoseria in space radiation, there was more DNA damage in HDAC4, and therefore an activation of the DNA repair pathways. HDAC4 was upregulated in the space sample as compared to the ground sample. So all of this to say is that the genes of interest are significant contributors to radiation and DNA damage response. But this begs the question, how are MORC2A and HDAC4 related? After investigating a literature review on the relationship between MORC2A and HDAC4 on cell senescence, we discovered the following process. As referenced in the image, P21 transcribes to cell cycle arrest. MORC2A recruits HDAC4, and together they repress the transcription of P21, which causes cell proliferation. This can help promote healthy muscle cells during radiation-induced muscle atrophy. To support our current research, the StringDB protein plot shows a connection between CDKN1A, the alias name for P21, and HDAC4. Through the previously listed bioinformatics pipeline, we discovered this unique pathway and the correlation between our identified genes. For our proposed experiment, we hope to further analyze the pathway and its future application. We will be describing our hypothesis and aims for our proposed experiment. We have two aims. The first one is to measure the effects of knocking out the transcriptional repressors MORC2A and HDAC4 on cell senescence, and the second one is to measure the effects of knocking out P21 on muscle cell proliferation. So for our first aim, we would use the C2-C12 muscle cell line, and for our experimental group compared to our control group, we would have an HDAC4 and MORC2A knockout with siRNA. They would then be irradiated and then stained with BGAL for markers of cell senescence, and then we would run a flow cytometry analysis on them. So for aim 2, we would have the exact same setup, except that the experimental group will have a P21 knockout instead. Then we would irradiate it and perform an EDU assay. And then we would dye it with Alexa floor dye and Host dye to look at the newly proliferated cells. Now I will be talking about the significance of our project and why it's important today. Previously, we have only looked into microgravity's effects on muscles and not space radiation. And this can be extremely detrimental because muscle atrophy is such a prevalent issue. First, muscle atrophy can lead to long-term health risks, such as arthritis and cardiovascular disease. This is especially important as NASA conducts longer duration human missions. Next, astronaut performance will be diminished because of decreased muscle strength, which can hinder crucial tasks in space. Lastly, muscle atrophy can cause re-entry issues, as weak muscles will result in difficulties during reorientation and landing procedures. Our study is not only relevant in space travel, but also for humans on Earth as well. For instance, skeletal muscle cells in cancer patients are often exposed to high doses of radiation as a side effect of chemotherapy treatment, which often contributes to the inability for muscles to regenerate. Furthermore, muscle atrophy is present on Earth due to aging. 50% of people over the age of 80 have sarcopenia, which is the loss of muscle tissue as a natural part of the aging process. Thus, our experiment provides gateways to further research on possible therapeutics. So if we target HDAC4 and MORC2A or P21 because they play a significant part in the cell senescence pathway, we have the possible implications of mitigating muscle atrophy and deep space radiation, as well as promoting human health on Earth by addressing age-related and radiotherapeutic treatment induced muscle atrophy. Here are our selected references. We would like to thank the people that have invested their time into helping us. Thank you to the Gene Lab for High School's team, Dr. Elizabeth Flavor, Ms. Jennifer Claudio, as well as our other mentors. Thank you so much for listening to our presentation. And here's our contact information in case you want to reach out for any inquiries.